Hello and welcome to another Polestar Pilates Hour. It's good to be with you today. Today it's just me, and we're going to be doing a little science work today, talking about human physiology. It's a new series that I've been putting together, so we'll wait for everybody to get in. We have a big enrollment today, so I hope I can live up to it. Please let us know always if this is something that you like, if it's helpful to you. I know that we had some really good feedback on the joint session with Shelley and myself, and we'll be doing some more of those and looking at different topics. Hopefully you felt like there were things that you could apply immediately in your daily practice of Pilates therapy and movement for yourselves and for your clients. Well, this is a topic that I've been teaching for the last 4 years in the university, looking at not just physiology, but pathophysiology. And if you've talked or heard me speak in the last couple of years, you've heard me also refer to pathokinesiology. And that's a course that I'm developing for online that it will be probably about a 10 to 20 hour course. I'm about a third of the way through. But a big portion of it is the physiology, and particularly the physiology as it pertains to movement. So how is it that we understand the science of movement if we don't understand the basic human physiology and understand where physiology comes from? What does physiology mean? Why do we need to study physiology? So these are all questions that we ask ourselves quite often in the sciences, trying to understand better what is the purpose of these sciences. So if you went through the Polestar training, we often talked about each of the principles as they related to the current sciences. So we talked about anatomy, physiology, motor control, biomechanics, energy medicine, communications, and psychology. So we brought those sciences into each of the movement principles to better understand how they influence, impact, and affect good movement, how they prevent injuries, how we can understand how to teach movement better, how we might identify disease and pathology. As a pathokinesiologist, one of the things that I am so excited about always is understanding first what is normal movement, what is normal physiology, what is normal motor control, so that when I see the pathology come in as a physical therapist, to me what's important is how does that pathology impact movement? Very seldom do I ever treat pathology. My job is to treat the effect of pathology or to influence function in the midst of pathology. Somebody comes to me with multiple sclerosis, I'm not going to be prescribing medication to treat the multiple sclerosis, but I will be teaching them how to re-educate themselves to move, how to have better function, how to increase their energy efficiency as it pertains to movement. So pathokinesiologist is defined as anyone who treats movement that has been affected by pathology. We could also use another term that I like, which is performance kinesiologist. And a performance kinesiologist is looking at how do I use kinesiology or science of movement to be able to enhance movement or enhance performance. So today we're looking at a very basic part of movement physiology. Right now I have it broken down into 3 parts. Putting this particular presentation together, I realize I probably need to have 6 parts. So we'll get back to you and we'll take it one part at a time as we go through as to what is a proper amount of content that somebody can take in. Now, the other thing that I'd like to know from you today, and particularly at the end of the day, is let us know if this is the kind of content that you feel is beneficial. Again, we like to mix it up. So things from cueing, things from energetic work, Pilates work, case studies, and now we're introducing some basic sciences to help enhance and improve all of our ability. I have to be the first one to admit that every time I study to prepare for a webinar, I think I learn the most. I get the most out of it studying and reading the articles and the books. Today, I'm drawing from 2 books. They're going to be listed in your post-production. One is Human Anatomy and Physiology, and the other one is upside down, but it's Human Physiology by Silverton. And Human Physiology by Silverton is the one that I use at the university. So these are solid books. Again, it's always nice if you want to start building a library and have basic sciences that you can refer back to. Doesn't matter what it is or what you see. If you need to understand something a little bit better, just like me, I go back to the books. And these are common sciences that have plenty to do with it. So let's jump right in. And this first slide is the objectives. And I take objectives seriously. If you've ever been on a course with me, you know that I feel like objectives are my contract with you and your contract with me to make sure that we're expecting and delivering the same things. So here I put in understand the somatic nervous system. So we're going to start off with that, looking at somatic means body and motor means movement. So we're looking at the nerves that are going away from the central nervous system to the muscles, our somatic motor nervous system. So we're going to talk about that. How does that conduction or message get to the muscle? And then we want to understand how the skeletal muscle generates power. So what is it? What's going on inside our muscles that is happening at a chemical level, at a hormonal level, at an electrical level to be able to generate torque? We're going to explain how the neuromuscular junction works. I think that's really important, of where the nerve actually connects to the muscle and the neuromuscular gap there and what takes place. We're going to understand the journey of the message from the spinal cord to the muscle. So that's the bigger picture. And then we'll look at different types of muscles as we get into more and more functional aspects of things. We'll look at a little bit at metabolism. Where does the energy come from to drive the muscle? And then we will describe different types of movement. And this sort of prepares us, if you heard me in the very beginning, I'm going to be teaching a course in person called Open the Gate. And that then starts looking at things like rhythmic movement and volitional initiation of movement, that it starts becoming important to understand how do these muscles, how do these nerves work in movement. So that's, if everybody's good, I get a thumbs up from you that we're okay on the content material. You let me know if it's too hard, too easy. You want more challenging, you let me know. Also, you have the Q&A button down the bottom. So you can Q&A me you can question me or answer for me at any time during the lecture. It's just us today. So nobody else is joining us. We're just going to hit the science. So here we go. So first, we're going to look at the somatic nervous system. And the somatic nervous system also includes the autonomic nervous system. I'm not going to spend a lot of time on that. But by its name, autonomic means that it's automatic. It's something that is subconscious. It comes from the brain stem, particularly the hypothalamus. And that is where we store our sympathetic and parasympathetic messages that are going to be stimulated through the parasympathetic and the sympathetic nervous system. Those also are efferent nerves, meaning that they're going from the central nervous system to a target tissue. So these are some words that I'm going to be using. So we have somatic, meaning body, the body's nervous system, peripheral nervous system, not the central nervous system. And we're particularly looking at the efferent nervous system, meaning going away from the central nervous system to target tissues. And more particularly, we're looking at the motor control or the motor aspect of the somatic nervous system. So we're looking at muscles being stimulated by these nerves that are coming from the body. And it's important to understand the autonomic nervous system does not stimulate skeletal muscle, but the somatic nervous system for muscular or motor is only muscle. Okay, so we'll take a look at that. So if we look at a big picture of this, we basically know that the central nervous system is the brain and the spinal cord. So when I talk about the central nervous system, I could be talking about either. It also includes the brain stem, the cerebellum, anything in the central nervous system wrapped in the dura. The dura is the connective tissue and fascia that surrounds the central nervous system. So sometimes you'll hear words like epidural, meaning that they're going to bathe around the dura of the spine if you were having a surgery, or the dura layer around the brain that they have to cut through to get to the brain in a brain surgery. So the motor cortex is the part of the homunculus, we call it, that has the map of the body on there, usually, and it represents the opposite side of the body, is where commands come from volitional movement. Like I want to bring my hand to my mouth, and the message from there comes from a motor plan that's in my motor cortex, and it's sent, and it sends messages down the spinal cord. And then down here at the bottom, I'm going to bring my little cursor on here so you can see what I'm talking about. But right here is the anterior horn of the spinal cord. And the anterior horn cells then are going to be responsible for that somatic motor nerve going to the muscle. Okay. So what we're going to be talking about today, primarily, so you get a pen here, primarily we're looking at this area here that we're going to be talking about. So we're looking from the anterior horn cell to the muscle and what's happening inside the muscle. That's going to be our primary focus. But understanding that volitional movement is coming from the brain. And this is why you've heard me say so many times that the more subconscious meaning it's happening below the brain, right? The more subconscious it happens, the more of a motor plan that it is, and it's just a command that goes down to move, the faster and more efficient the movement is. The more cerebellum involved in the decision-making of movement, the slower it is. So if I give you a voluntary command or cue of pull your navel to spine, you have to basically organize that thought in your head from the ears listening to me, and then pull your navel into your spine. Where if I just push on your shoulders in a certain way, your transverse abdominis is going to automatically contract. That pressure contraction is happening from down here in the spinal cord. So something's going to start making sense. I'm going to try to keep tying them into concepts that we've discussed on a much more global scale of why it works like this in the physiological and the cellular level. Shall we move on? All right. So when we look at this efferent motor nerve, it's coming down from the spinal cord, okay? So just giving you that reference, it's coming down from the spinal cord, and this message is traveling in this direction, okay? And what these little myelinated sheaths are right here, those create a saltatory effect. Saltatory, if you think of Spanish, saltar, is to jump. And so it speeds up the nerve conduction. So these nerves are very fast nerves, very big nerves, and they conduct what's called an action potential down the nerve, okay? And what an action potential means is that it's a signal. It's a message, right? And the message is coming down, and the message is going to release something here at the distal end of the nerve into its target tissue. Now, an efferent motor neuron's target tissue is always going to be muscle. See a comment here from Bernie. It says, is this another way of talking about spinal reflexes and feed forward system? Absolutely. And we're going to go there, Bernie. So I appreciate the question. So as this action potential travels down the nerve, and what's happening in action potential is that we have a resting membrane potential in the nervous system. And it is determined by the amount of sodium on the outside and the amount of potassium on the inside, right? These are ions. And that's why things like having enough calcium in the body, and you'll see why it's so important to have calcium in the body, not just for bone health, but also for nervous health, muscle health, brain health and integrity, really depends on calcium. It's so important to us. And as well as sodium and potassium. So sodium, potassium on each side of this nerve wall, cell wall, and it is at a negative 70 millivolts. That's its resting membrane potential. So what happens is, as there is a stimulation that comes down from the reflex that Bernie talks about, a stretch reflex coming from another muscle or the same muscle, saying that there's a load and that load has to be dealt with. We need more muscle fibers. Sends a message down that nerve, that efferent nerve, and that action potential is called a depolarization of the nerve. Depolarization means that it's going to go towards the positive. Remember I said it's a negative 70 millivolts? It's going to move up towards like negative 40 millivolts. And so what's happening is that sodium and potassium are passing through the cell wall as that action potential comes down. and as well as the saltatory feature. And it's going to come down into the muscle fiber. Okay, so we've talked about action potential is a depolarization, meaning a change of ions, an electrical charge coming down the nerve with a message to the end of the nerve that's going to communicate with the muscle. That's the next thing we're going to look at here. So here we see a picture, and this is considered the neuromuscular junction. This whole thing is the neuromuscular junction. And what we're looking at here is this is the end of the axon of the nerve, and this nerve impulse is coming down. And as the action potential gets down to the end, you'll notice here's our calcium. And calcium now goes inside of the membrane. And inside here are these little vesicles, and these vesicles contain acetylcholine. And acetylcholine is a neurotransmitter that is going to send a message to the target tissue. So these are important words to sort of play with and understand. But target tissue here is the muscle. The nerve is the messenger. And the messenger is done by a depolarization or action potential that stimulates calcium to go inside. The calcium then stimulates these vesicles right here to release the acetylcholine into the cleft. So the cleft is the tissue, is the space in between the nerve and the target tissue, which is the muscle. Now on the muscle side, it's a little bumpy and uneven with a lot of receptors. And these particular acetylcholine receptors are referred to as nicotinic receptors. You don't have to remember that, but that's the name of the receptor. And it's just waiting for the acetylcholine to come inside these receptors. And once the acetylcholine hits the receptors and there's enough acetylcholine in the cleft, it's going to initiate another action potential that's going to go down inside the muscle into the T tubules. So it's like we have, and all the action potential means is that it is an electrical message. So the electrical message coming from the spinal cord down the nerve releases the acetylcholine. Acetylcholine stimulates the target tissue and the receptors that initiates another action potential or a depolarization, another word for that, of the muscle that's going to bring on a contraction. So again, just thinking from the top down, going from the anterior horn cell of the spine, there's an action potential, a message coming down. It's going to allow the calcium to go inside the end of the axon or the cell. It's going to then stimulate these vesicles filled with acetylcholine to do what's called exocytose. Exo means exit. It's going to leave the cell. Excuse me. It's going to leave the cell and connect with the receptors. The receptors are going to start another message that's going to go down the muscle. I think we're good up to that point. Okay, so that's the somatic motor nerve that we're looking at all the way to the neuromuscular junction. So now we want to move into the muscle physiology from that stimulation or from that action potential. And we can look at the muscle skeletal physiology. So the first thing that we notice is that the muscle is the large, like if we were dissecting the body, we pull the bicep muscle out with its tendons. It's a large muscle. But really what it is a lot of muscle fibers, and inside the muscle fibers, the myofibrils, and inside the myofibrils, the filaments that actually contract. So there's a lot of activity happening here. If we look at the big muscle, right? If we look here in this picture, this is you see here the muscle, and inside the muscle is the fascicle, and inside the fascicle is a muscle fiber cell. This is where the cell is, and it has a nucleus. And inside the muscle fiber is what we call the sarcoplasm. And that is where the filaments lie that are responsible for contraction. We're going to go through each of these so we understand them a little bit better. All right. So inside these filaments here are what we refer to as the actin-myosin filaments. And the actin-myosin filaments are thought to be, it's a theory. There are some people who dispute the theory in physiology, but for all intents and purposes, this is our best understanding of how the contraction and the torque actually take place. We're going to go into a little bit of depth with each of these. So if you have any questions on these, feel free to put them up. You're not going to be the only one with questions. So if you misunderstand a term that I'm using, or you see something up here, let's make sure we clarify it and get you so that you have good sound information from the nerve all the way to the muscle contraction. So here, what we're looking at is that nerve is inside the fascicle. And you see this yellow, this is the nerve, and it is stimulating the muscle fibers. And so one nerve could actually stimulate a lot of fibers and create an action potential. And that's what we're going to look at in this next slide, is we have what's called our T tubules. So the T tubules are what you see in the yellow. And these T tubules are what conduct the action potential that came from the neuromuscular junction. So remember, release the acetylcholine into the cleft. The receptors of the muscle picked it up, and they're sending an action potential down through the muscle. So all the muscle is receiving this action potential. And this action potential is going to send a very important message all the way down to the fibrils, and it's going to communicate. Now, there's another piece of anatomy here that's very important in the muscle, and this is the sarcoplasmic reticulum. Sorry, guys. Another very important part of the anatomy is the sarcoplasmic reticulum, and the sarcoplasmic reticulum contains no other than calcium. So it has calcium in here, and just a bunch of calcium in here, right? And what happens when the action potential comes in, the action potential actually will stimulate the release of the calcium from the sarcoplasmic reticulum. The calcium surrounds the filaments, and I'll show you in a second how it actually facilitates a contraction. While we're thinking of that, I have a comment from Sarah Edwards. Hello, Sarah. Given this information, there are a lot of considerations for nerve damage or neural pathologies, correct? And the answer is absolutely correct. And this is why, if we understand normal physiology, then we can understand when somebody says, oh, Lou Gehrig's disease or ALS affects and kills the anterior horn cell of the motor units. So now you can go, oh, this is a peripheral nerve disease. It's not going to send an action potential to the muscle. And so the muscle is going to be paralyzed. It's not going to get the stimulation that we think it should get, right? So the better we understand these, when somebody talks about, there's a calcium deficiency, and therefore there's a fatigue, there's a syndrome that causes fatigue. When we go into connective tissue, we're going to be able to identify different connective tissue problems. So the take-home message on this slide is that depolarization is traveling down these T tubules. So it's going down the T tubules, and it's going to connect with these, the T tubules to the myofibrils and facilitate release of calcium that bathe the filaments and facilitate a shortening of the myofibril. And this is where it gets really interesting to me. So if we look here, we can see the big picture. I know that repetition helps a lot, but if we look at this, we say, okay, we have up on top here, this is the nerve coming down, and it sends a message to the muscle. Right here, we see the ACH being released. That message then is going to travel down the T tubules to the sarcoplasmic reticulum, which is going to release the calcium into the space around the filaments. And in the filaments, we have two main components, the myosin and the actin. The actin slides on top of the myosin by the head of the myosin pulling it across. What the calcium allows it to do is it allows the head to bind to the actin. And that's by its binding to what's called troponin. You don't have to remember that name if you want to get deep into the physiology. But the troponin is necessary to be attached with calcium so that the head can attach to the actin And then ATP, adenosine triphosphate, our main source of energy for almost everything we do, is necessary to be able to pull that pull the actin across the myosin. And that then shortens the myofibrils, and millions of myofibrils being shortened will shorten a muscle and cause a contraction. How much? Enough, right? We often talk about the subconscious contraction is going to be enough. Its job is to anticipate and contract the right amount of myofibrils with this methodology. Now, what I want to make clear here is once you understand this physiology of the muscle, it is the same for every type of movement. All stimulation of muscle is done this way, even heart muscle, right? This is how muscle contracts. So it might be a little overwhelming at first, but understanding is where we go like, okay, I understand it now. When the filament slides together, the fibril shortens, the myofibril shorten, the muscle shortens, and now all of a sudden we have a contraction. All right, let's go a little bit deeper, one more deeper on here, and then we're going to bring it back to a more global idea. Before I go there, Gwilette had a question, is what if someone has neuropathy? Now, neuropathy is typically a sensory deficit in the nervous system. So our body has afferent nerves. Afferent means it's going towards the central nervous system. Efferent means it's going away from the central nervous system. Okay, so an afferent problem of a sensory nerve could be causing neuropathies. Neuropathies could be pain. Neuropathies, typically pain or numbness, where the peripheral nerve is not communicating well to the brain, or it's not getting good information. So you have people that have diabetic neuropathy in their foot. That is a sensory nerve problem. And today we're talking about motor nerves. So motor nerves are going away from the central nervous system, and then sensory nerves are going into the nervous system. They're both incredibly important. Neuropathy is an incredibly important pathology that we can deal with by decreasing the sensitivity and decreasing the body's reaction. For example, a neuropathy could cause either muscle inhibition or excessive muscle excitation, a spasm, or flaccidity or weakness based on that information coming in from the body incorrectly. So we'll talk, just making me think I need to talk about the efferent system as well. It's just not going to be today. So let's move to the next slide. And this one sort of brings a lot of it together technically. And if we look at, I'm going to use a little different feature here so that we don't get too distracted. But here we have the T tubules and the T system coming into the sarcoplasmic reticulum, right? So what ion is inside the sarcoplasmic reticulum? Calcium, right? So then we can go over to the next thing and look a little bit deeper, and we see this idea of the sarcomere. And here, this midline is the support of the myosin fiber. So we're going to come back here and look at how these fibers here, in particular, the actin is on the Z line, and the midline or the band is where the myosin connects. So you can see how the actin slides over the myosin and can shorten the actin myosin. This is the striation we see under the telescope of the actin myosin. Let's go back here. My apologies. I want to go to this one. So now we can look here and we can see how this is a lengthened sarcomere and this is a shortened sarcomere. So if we were to take the measure, we would notice that the measure of the myosin stays the same, right? We can see that same thing here. This is open on this side, closed on this side. You can see how the green has slid all the way across here. And then for this to happen, right, the calcium has to be released in this area. And we can zoom in on this as well here, right here. And here we see that the head of the myosin are pulling the actin across it, pulling it in this direction. I guess I'm going to use the pen here just to make it a little bit easier. But it's pulling it in this direction. And it's closing that space down to make a more tight contraction inside the muscle. So if we go back a slide, we sort of see this again. Just to reiterate, nerve, muscle, global muscle, right? Comes in and you have the neuromuscular junction. The nerve sends a message with the acetylcholine as the messenger. It's received in the nicotinic receptors. It causes another action, electrical action potential, to go down the T tubules. That opens up the channels for calcium to flood the actin myosin filament. The calcium causes the head of the myosin to attach to the actin, and ATP then causes the actin to slide over the myosin, and that's the contraction. Okay, so there might be a question out there, which is, what is ATP, and where does ATP come from, and why is it so important? Hey, Kimberly Garlick, my dear friend and senior educator in Australia, asked, is that the same as the sliding filament theory? And yes, it is. That is the sliding filament theory. So let's move into talking a little bit about ATP. So ATP is considered a basic energy source for the body. It's the formal name is adenosine triphosphate. And what happens is that anytime that it is causing a transport of anything, right, it is giving off a phosphate. So what we need is, in our food and our metabolism, is to keep bringing new phosphates to the blood, through the blood, to the cells, to be able to have enough P, right, have enough phosphate to make ATP after it's been used up and converted to ADP. So the phosphate keeps creating energy. It needs to come in through that mode. Now, typically we talk about glucose in the blood as the primary contributor to the phosphate. Creatine phosphate is a byproduct of glucose. And so we see the food comes into the body right here, and that gives off a phosphate. That phosphate comes over and attaches onto that ADP. And now we have adenosine triphosphate, right? And that's going to be the energy. And as it hydrolyzes, it's going to give off a phosphate and convert back to ADP. And this cycle continues over and over and over again, known as the ATP cycle, right? The phosphate cycle. And our body continues to convert glucose into phosphate for energy. So what happens if we don't have enough glucose, right? Well, then we fatigue. And the fatigue basically says we're done with that, right? That activity. We get tired, or we need more fuel. So if you're running a long distance race, you're taking food in as you're running that race to provide more and more phosphate through the glucose in the blood to that body. Now, what happens if you have too much glucose in your blood? Well, that's not healthy either. Insulin gets released, which is a hormone, from the pancreas, and the insulin then is going to store sugar in the liver and possibly fat if it sits there long enough, right? And then when we need more sugar, the glucagon is going to be released, and there's going to be more glucose in the bloodstream based on our need, right? So it's always managing the equilibrium, or the homeostasis, we refer to it, of the blood sugar levels. And our blood sugar level should be somewhere around 90. If it goes much lower than that, that means we're not getting enough phosphate to our body to do its job. If it's doing more than that, then what ends up happening is we have excessive glucose in our blood. And that's where we talk about metabolic disorders like diabetes, type 2 diabetes, or other types of resistance to insulin disease in our body. How we doing? Okay. Can I get a thumbs up if we're giving decent information here? If this resonates with you, if we're connecting some dots would be good for me to know about now. While you're doing that, I have another question from Gwilette, and she says, does intermittent fasting have a negative effect on the muscles? And the reality is it's quite the opposite. Intermittent fasting has been shown to do a much better job of managing your energy source. And we could talk about that another time. I will refer you to a great, you've heard me talk about Andrew Huberman and Huberman Lab, and he talks a lot about intermittent fasting. A number of other people do as well, but those are good resources from scientists who really study intermittent fasting. But everything that I've seen are intermittent feasting. I like to think of it as intermittent feasting because I like to think of the eating part, not the fasting part. But the intermittent feasting really does teach your pancreas and your liver and your hormones a better way of processing and storing glucose. And so people who do intermittent fasting seem to have better access to glucose at the right time and the muscles. So we tend to eat too much and we don't give the body enough time to process it before we're eating again. And that can be tough. So again, I'm not a nutritionist. It's not my area, but I do get the fact of what has to happen at the cellular level, and that's important. Thank you for your feedback, everybody. Let's keep going. So here we look at different types of muscles and how they work. And the way that I like to break this down is you have the 100-meter sprint, you have the 400-meter sprint, and then you have the 1,500-meter run, right? So if we go backwards on this, the type 1 fibers, let me switch to a laser here. The type 1 fibers are considered slow twitch and oxidative. So what oxidative is referring to is that they are aerobic. So when you hear people say aerobic exercise, what you're doing is trying to go long enough, but at a low enough intensity that the body is oxidating, right? Aerobic oxygen, fat in particular, but also carbohydrates, and is able to create a slow, steady burn of energy at a sub-maximal level of performance. So this type of muscle behavior would be best for more of an endurance activity. I'm going on a walk for a long time. I'm going to go hiking. I'm going to go swimming. Might be using more slow twitch oxidative fibers. Their contraction speed is slow. They fatigue slow because they have lots of oxygen, lots of blood, lots of glucose in there, so they last a long time. Their diameter is small on purpose. So we could think of muscles like the multifidi in the back. We could think of muscles that are like the rotator muscles around the hip and the shoulder. We could think of deep abdominal muscles, maybe the transverse abdominis, as being more of the oxidative slow twitch muscles. They're meant to work all day long in supporting us in our position, right? They are, let's see, high mitochondrial concentration. That's so that the oxygen can be metabolized. And then their glycolytic enzyme concentration is low. So that means how much creatine phosphate is inside the blood there that's bathing it. It doesn't need it. It has so much blood. It has plenty of energy coming into it for the level of work. Now, the type 2A is a mixture between something new that we talk about, which is the glycolytic with the oxidative, right? So what that means is that this is like the 400-meter run. I don't know if you've ever run the 400-meter, but typically when I ran the 400-meter, I hit that 300-meter mark and I died. Like it just like there's no energy left, right? So that's a very good example. We'd have to condition ourselves to have oxidative as well as the creatine phosphate or glycolytic phase in the whole 400 meters. Somehow you got to be able to convert. Now, I'm very much on the type 2B kind of body make, and that is that it's fast twitch glycolytic. So the contraction speed is fast. I was a pole vaulter. I like to jump. I like to sprint. Didn't like anything longer than 200 meters. Why? Because I didn't like using these muscles, I guess. I was much more adept in my childhood of using type 2B, fast twitch glycolytic fibers, right? Now, as I age, I find myself in activities that are using much more of the oxidative. I get on my bike for half an hour, I go for a hike, I do some Pilates, I work on some core control, I work on some body awareness, and I spend a lot more time in here than I do over here now, okay? The idea of the glycolytic enzyme, very fast. This is our 100 meter, right? So in 10 seconds, the creatine phosphate, there's enough ATP or phosphate inside the muscle, inside the fibril that we're talking about, to be able to give a max exertion for around 10 seconds, right? And so that's our sprint. So if you think of muscles like the quadriceps, the glutes, the hamstrings, the calf muscles, the gastroc, they're going to have a high percentage of type 2B fibers in them in somebody that is a sprinter. Versus somebody who runs distance, you're going to find a higher percentage of slow twitch oxidative fibers or something in between with a type 2A. So it just depends on the activity and the nature of the muscle. It'll be interesting to see as we become more and more of a blob society of sedentary people, what's going to happen to our mixture or blend of type 1, type 2A, and type 2B muscle fibers. Bernie had a question. Do you find that slow twitch muscles have more muscle spindles per square inch? Yes, at least 6 times the number, Bernie. When you think of the multifidi muscles in our back are short muscles that usually span 3 or so segments. And those fibers have 6 times the number of muscle spindle fibers. And you might ask, what's a muscle spindle fiber? Well, that's part of a reflex, which we're going to talk about in just a second. But a reflex is, let's say, a muscle stretch reflex. There's a little stretch on the muscle that's saying that the load is too much. I need more muscle recruitment. And it's then going to send a message to the central nervous system that it needs more muscle. Well, if you think of posture muscle, what's holding our body up? What's keeping the ability for us to balance our spine on top of our pelvis? It's a really weird kind of physics that we can walk as bipedal animals with a bicurve spine. So what has to happen is very fast, there have to be adjustments in the spinal muscles, especially the deep spinal muscles. So for that reason, there are a lot of receptors inside those muscles called spindle fibers or muscle spindle fibers. And this alpha gamma coactivation that happens inside these little muscle fibers basically tell the spinal cord, the central nervous system, not necessarily the brain, but the spine, that it needs to adjust. It needs a little more stiffness here. It needs a little more relaxation here. So it's always adjusting, Bernie. And because of that, it needs more muscle spindle fibers. Where if you think of a muscle that's more responsible for an acceleration, right? Like we're running and we're sprinting and we're using the gastrocnemius, is going to have less muscle spindle fibers than, say, the soleus, that is a deeper calf muscle that we use more when we're standing. And it's just sort of maintaining stability on top of the ankle. Also, we know that these same messages that come in the spinal cord also do travel up, and they travel up into the cerebellum that are responsible for proprioception and balance, where we know where our body is at all times, where our fingers are up or down, our feet are up and down, what we're standing on. So very interesting discussion. I think we'll come back on the next one and we'll talk a little bit more about reflexes and also sensory. I saw some people interested in neuropathies, so we can talk a little bit about that. And I want to take some time to talk about pathologies of the somatic nervous system and the muscle itself. So we can start with some of that today. We'll have a little bit of time, maybe 5-10 minutes afterwards. Kimberly Coop said, is this a good reason to do more HIIT work? Can you just expand for me what HIIT work is? I'm so bad with so many acronyms, I always forget, but maybe I do know what that is. And then Sarah Edwards said, so are different body types naturally prone to different muscle fibers, or can we train the muscle types to develop regardless? And that's an age-old question in muscle physiology. Are we born? Is it more genetic that we have more fast twitch fiber than slow twitch fiber, or is it environment and nature, that environment that trains us to have more or less? What we do know is that as we age and we train, we tend to have a tendency to convert the type 2B or type 2A fibers, particularly type 2A, into fast twitch fibers or slow twitch fibers, sorry. So it makes sense. As we age, we tend to go more towards an oxidative state, more endurance, less explosiveness or acceleration. Let's see what Kimberly came back and said. High intensity interval training. Thank you for that clarification. Each kind of training that we do will have a different response. So for example, if we're doing plyometrics or isometrics, or we're doing short repetitions, high load, or low repetition, or how do I say that right? Low repetitions, high load, like powerlifting to actually stimulate large muscle recruitment, or light load over long repetition or endurance to be able to have a little more power and endurance in the muscle will change the way the muscle fibers react. So that's sort of a cool thing. All right, let's jump into movement now, because you guys have been very patient with me on this, and I hope the science is giving you some good foundation for us to continue to build on as we talk about more and more pathology and movement and things that we see out there. What I wanted to finish up with today is this particular slide looking at types of movement. And the first thing that I want to explain here is that everything we've talked about up till now happens in every one of these, all right? Because from the somatic motor nerve down to the muscle, it is the same physiology for every contraction, no matter what type of contraction or what type of movement it is the same. So very important for us to understand that. So the first type of movement I have here is reflex. And this is something that's age-old. In science, the scientists of the 1800s and early 1900s believed that all movement was a result of reflex. So a reflex, for example, you step on something sharp or hot, your foot picks up, and the other foot straightens out to keep your balance so you don't fall down. Or if I tap your tendon, it stretches the muscle spindle fiber, and it causes what's called a jerk reflex. So the knee jerk reflex, the bicep reflex, the tricep reflex. It tells us if there is normal nerve conduction to that muscle, that there's not a problem in the spinal cord or in the central nervous system. So when we do our jerk test, we're looking at reflexes. And these are the most basic of basic. They basically control a sensory input and has a reflexive response, an automatic response. So you might think of things like coughing, like I was just doing. If something gets in my throat, I don't think about coughing. I have an automatic reflex to clear my throat. Or if I get dust in my nose, a sneeze is a reflex. With children, when they have problems developing, we might put them on top of a ball and roll them side to side to try to get their head to start doing what we call a righting reflex. Also, you can put a child's face up next to soft skin and they'll start rooting. It's a reflex that ensures that a healthy child is going to nurse or is going to eat or drink from a bottle. So these reflexes are the most basic, they're the least complex, and they're inherent, meaning we get them from birth. They're just part of our survival in our nervous system, right? And they're rapid. Voluntary movement is quite the opposite, especially for us as humans. We rely on external stimuli and it is at will. So we look at something, or I want that, I want to locomote over to that point, and my body has to try to figure that out. Or even more complex would be things like typing on a computer or playing the piano or a musical instrument, playing sports. All of those things are learned through a voluntary system, which means the higher central nervous system is involved. We see it, we hear cues, we try to do the movement, and it's real awkward. We recruit too many muscles. It's a novel movement, a co-contraction. And then as we refine that process through practice, we convert that voluntary movement to more and more efficient movement. Super cool. It's by far the most complex and relies a lot on feedback mechanisms, feed forward, feedback mechanisms to be able to successfully perform a task. So when we typically think of voluntary movement, we're not thinking of muscles. We're thinking of tasks. This is what Shelley and I were talking about last week and a couple of weeks before that, is how do we cue good core control? But we don't cue good core control by telling a muscle to contract. We tell it by the task that is at hand. Does it maintain, does that task, that body respond by maintaining proper alignment and organization efficiently between the head, the thorax, the pelvis, and the extremities? That's core control, right? And so voluntary learning of muscle contraction is not the most effective, but voluntary learning of tasks. Like, for example, how many of you have learned to play the piano? And if you learn to play the piano, you weren't telling yourself, okay, I'm going to flex my flexor digitorum, the left hand, and do the lumbrical extensors of the fourth digit to be able to put my finger down onto that key. We didn't do that. We watched, we tried, we failed, we learned from the cue from our music teacher or watching somebody else play, and we started learning how to do those movements. We never once considered an isolated voluntary muscle contraction. I think this is really what sort of spurred me to say, let's teach physiology so people understand why cueing a muscle doesn't make any sense through an isolated muscle cue, where doing a muscle cue through movement and through a task could make all the sense in the world of how we do the task, or like playing the piano. Now, there's one more type of movement that we refer to as rhythmic. And this is an interesting blend and more applicable sometimes in animals. For example, we could take a cat, and if you spinalize the cat, meaning you sever the spinal cord down below the mid-spine, the cat can still walk on a treadmill. If the treadmill is moving, the hind legs are going to move with the speed of that treadmill. Now, humans sometimes have a little bit of that, and that's what we refer to as the central pattern generators. These are located in our spine, and through a lot of practice, things like walking, running, swimming, marching, these are repetitive movements. There is an initiation voluntarily to start that movement often, but then there are central pattern generators that keep it going. You never have to. Could you imagine having to think of what muscles you're using for running while you're running? Could you imagine even thinking of when you're supposed to inhale and exhale when you're running? Do you exhale every time the foot hits the ground? You'd be breathing, right? Well, some of us do that anyway when we run. But the idea is that these rhythmic patterns are patterns that are learned, and they become automatic to some extent. It's not a reflex. We're not voluntarily learning it at this stage of the game. It becomes more automatic and rhythmic. It's a rhythmic movement. So pretty cool stuff. We're going to come back and talk a lot more about this. As a matter of fact, the course that I have coming up soonest is going to be Open the Gate, as I mentioned early on. And we're going to be talking a lot about walking and running and how the nervous system responds to the load and how we can condition and cue proper organization for running and walking. So I hope you join me, and some of you might make that trip to come to the farm finally in the fall and come spend some quality time with us here at Southwind Farm. So this concludes the lecture for today. Again, just reiterating action potential, neurotransmitters released in the cleft, nicotinic receptor initiate, another action potential in the T tubules throughout the muscle fibrils and facilitate the release of calcium. The calcium then goes to the sarcoplasmic reticulum that releases calcium and bathes the actin myosin to facilitate a sliding contraction of the actin and myosin, right? It uses ATP, and the ATP is the energy that comes from glucose, the P, particularly the phosphate, and that is renewable depending on the activity, whether we are doing long distance or short distance or speed and max contraction versus long endurance of submaximal contraction. And this is why we ask people, what is it that you believe you should be participating in? If somebody tells me they want to be a long distance runner, then I'm thinking of working on more rhythmic movement, more type 1 fibers, looking at efficiency of their activities and building up their endurance and looking at their postural muscles, sort of what Bernie was talking about, the multifidi. How do I get those to stiffen appropriately for a rotostability of a bipedal animal running down the road for 5-10 miles, right? So pretty cool stuff. I hope you enjoyed it, and that we will continue with this periodically with the physiology of movement. And I hope that you enjoyed this material. We are going to ask you something. So just a thought here. Melissa will be sending out an invitation to you to maybe give a quick shout out, a videotape or audio tape or recording, maybe 15, 30 seconds even, of what you like about the Pilates Hour. And more specifically, if you like something particular about today's lecture, to do that and then just send it to Melissa. And we would love to promote you and have you promote us so that we can get free knowledge out to more people to make better decisions in how we move the world. And you know, our word this year is Movens Mundi, moving the world. We're celebrating our 30th anniversary in September. We wish you the very best. Hope that you are moving the world out in your neck of the woods. And most importantly, I remind you to be kind. We can all be kind, make a big difference. Well, next week, by the way, Shelley will be taking the programming. So you will be with Shelley for the Pilates Hour, and then I'll return the first week of September. So wishing you the very best, and we'll see you in September.